Okay, this is our third presentation on intracellular membranes, and we're going to focus this portion or this presentation on the signal hypothesis for protein translocation. Now, I'm sure many of you are familiar with this. You've had it either in earlier biology classes, was alluded to, I'm sure, last semester in biochemistry. And basically what this hypothesis states, we call it a hypothesis, but it's really a given explanation for what happens now. It's virtually universally accepted. But we talk about this in relation to translation of proteins that are on membrane-bound ribosomes. And it's used to explain and understand how nascent polypeptides get from the cytosol through the ER membrane into the lumen. And with modifications, of course, it can be used to explain how proteins get inserted into membranes. And of course, by membrane flow and vesicular traffic, it is the basis for how proteins get excreted from the cell. So a couple of key points here I've already alluded to and we'll talk about in more detail. We're going to talk about a signal peptide on a growing polypeptide chain. We're going to talk about a translocator apparatus. Of course, there are the ribosomes that are involved. We're going to talk about a signal peptidase, and we're going to talk about proteins going from the cytosolic face of the ER membrane into the luminal region of the ER, so those proteins have to cross the ER membrane. Now, we alluded to a common pool of ribosomes that are involved in 3 ribosome cycles. We're not going to talk about translation of messenger RNA on non-membrane bound ribosomes because you talked about that last semester in biochemistry. One thing I do want to highlight, though, is that this signal recognition particle cycle plays a role in getting proteins that initiate translation on free ribosomes and getting those proteins to continue translation on membrane-bound ribosomes. An important point that people tend to forget is that all protein synthesis, whether it's synthesis of proteins that are going to stay within the cytosol, whether it's synthesis of proteins that are going to be excreted from the cell, or whether it's synthesis of proteins that are going to get bound into membranes. All protein synthesis begins on free polyribosomes. And this signal recognition particle cycle is the intermediate cycle to get translation occurring from free polyribosomes onto membrane-bound ribosomes. This is schematically what happens. Let's put this in perspective first. On the bottom of the screen, we're in the lumen of the ER. On the top of the screen, we're in the cytosol. What this is depicting is protein synthesis is beginning on free polyribosomes. So you've got the mRNA molecule, the tRNA intermediate, obviously, to translate the mRNA. You've got the small ribosomal subunit, the large ribosomal subunit. Here is the signal peptide, the signal sequence on this newly synthesized polypeptide. And then here is a particle called a signal recognition particle. This signal recognition particle will bind onto the signal sequence. When this binding occurs, this causes a pause in translation. This pause in translation occurs because one domain of the signal recognition particle recognizes the signal peptide on the newly synthesized protein, but another domain inserts itself into the P site on ribosomes to cause a pause in translation. Something to keep in mind, there's a protein translocator sequence, conglomeration of proteins actually, that are in the ER membrane, and there's a signal recognition particle receptor complex in the ER membrane. Let me stop here just for a second. Do you remember in the last presentation we talked about rough microsomes and smooth microsomes, and we talked about the fact that you could isolate and segregate rough microsome membranes from smooth microsome membranes. And if you stripped off the ribosomes from the rough microsome membranes and then looked at the proteins that remain in those membranes, if you compared those proteins to proteins that were in smooth microsome membranes, you would get differences in the protein components of the rough versus smooth microsome membranes. Some of the proteins that are in the rough microsome membranes would be these protein translocators and signal recognition particle receptor proteins that be in the rough microsome membranes, not in the smooth microsome membranes. Now, anyway, one can imagine that these protein translocators are going to be in a configuration that must remain in a closed configuration so that calcium, which would normally be stored in the lumen of the ER, doesn't leak from the lumen of the ER into the cytosol. So these protein translocators are closed as protein synthesis is beginning on free polyribosomes. But now, lo and behold, this signal recognition particle binds to the newly synthesized polypeptide, causes a pause in translation. This pause in translation allows the signal recognition particle and the ribosome to bind onto a signal recognition particle receptor that is in the ER membrane. When this signal recognition particle is bound onto the signal recognition particle receptor, the signal recognition particle is released and translation continues. Translation is continuing as a loop of protein that's coming through this protein translocator. So the translocation is occurring at the same time the translation is now occurring, because translation, protein synthesis, has resumed. This translocator complex has been isolated. It's a multi-protein complex. It's essentially a water-filled channel. It's given the name SEC61. You don't really need to remember that for an exam, but this protein translocator, SEC61, is highly conserved in all forms of life, in all domains, in all kingdoms of life. So it's a highly conserved protein translocating machinery. Here is the ribosome binding to the SEC61 translocator complex. These are images that are based on EM reconstructions. So this is the ribosome. This is part of this SEC61 complex. So the small and large ribosomal subunit. And I think you might recall from biochemistry that as these subunits come together, they form a tunnel or a channel in the ribosome through which the newly synthesized polypeptide can translocate. Here is the SEC61 complex. It actually has a central pore in the SEC61 complex. This central pore actually lines up with the channel in the ribosomal subunits. The SEC61 actually consists of 4 complexes, and the polypeptide actually will pass through this channel in the SEC61 complex because the ribosomes will line up in just such an orientation so that the nascent polypeptide can go through this channel. Now, translocation of proteins across any membrane occurs simultaneously with protein synthesis. So there's no additional energy requirement for translocation beyond that which is needed for translation. So we say there is a co-translational translocation of proteins across the membrane. This occurs across the ER membrane, but it also occurs across bacterial cell membranes or archaean cell membranes, as proteins that are made in those prokaryotes have to get to the outside of, say, the bacteria. So this is an evolutionarily conserved process. So what it is saying is that translocation across a membrane does not require any additional energy. Co-translational translocation. A point you don't have to worry about. Sec61 is homologous to the complexes that are involved in translocating proteins across the outer and inner mitochondrial membranes. This is a little bit of the structure of the ribosomal subunit. I'm going to go through this relatively quickly because you don't need to know that much about the details. But here's the ribosomes, the small and large ribosomal subunit, the protein that's coming through the channel, the signal recognition particle. The signal recognition particle binds onto this signal sequence on the nascent polypeptide, and it also can hinge, and another domain can bind into the P site on the ribosomes to cause a pause in translation. A little bit more complex diagram, but basically the signal recognition particle consists of a small RNA molecule shown like this, and then 6 different protein subunits. Turns out that this conglomeration has several domains. There's a translational pause domain, there's a signal sequence binding pocket domain, and there's a signal recognition particle receptor binding site domain. And interestingly, this signal recognition particle binding site domain also has a GTPase activity. This is just a little bit of the ribbon diagram that biochemists can project with computer programs showing models of macromolecular structures, and then the signal sequence binding pocket. In some cases, proteins can translocate across a membrane after protein synthesis is completed. This can occur in some instances in the ER. It can certainly occur in mitochondria. It can certainly occur for peroxisomes, and can certainly occur for bacteria. If this translocation is occurring after translation occurs, that's going to require additional energy as well as other translocator proteins. So here's the model. Here's a protein being synthesized on free ribosomes. The protein is free in the cytosol, and now this free protein has to translocate across a membrane in a post-translational manner. And that's going to require different types of proteins. In bacteria, we know these proteins as the Sec-A proteins. Not something you have to remember for this course, but these Sec-A proteins, as they function to move a newly synthesized protein across a membrane, require ATP, so additional energy. In eukaryotes, there's proteins analogous to the Sec61 complex, groups of proteins. You don't have to remember the names, but Sec62, Sec63, 71, Sec72. These post-translational translocation mechanisms require utilization of ATP, and they require additional chaperone or binding proteins. So they require energy. Getting back to the signal hypothesis for protein translocation in the ER, the ribosomes bind, the protein translocates, the growing polypeptide translocates through the ER membrane, the signal sequence is bound into the translocator complex. There is a signal peptidase that may recognize this signal sequence. It will cut it or clip the protein there so that now you've got a soluble protein in the lumen of the ER. When this protein is clipped or cut, the translocator needs to close. When the translocator closes as a result of the signal peptidase cutting the signal sequence, the bound ribosomes will release themselves from the ER membrane. Look at this as a model. Soluble proteins translocate to the ER lumen if the protein is destined for secretion, you can imagine the protein translocates across, the signal peptidase cleaves the protein, and you've got a soluble protein in the ER lumen. This soluble protein will get packaged into a vesicle. The vesicle will flow to the Golgi, go through the Golgi, go through protein secretion, like we alluded to earlier, and in which we'll discuss in more detail a little bit later. How does a protein translocator become active and inactive? That's kind of an interesting question. Not going to ask you to be too overly concerned about the details, but this is the structure of the Sec61 complex. People believe that its conformation changes. It's almost like there's a plug on a spring mechanism, and that as a protein is being synthesized on ribosomes, that protein will push through and push this plug aside. As the plug is pushed aside, then the newly translocating protein can push its way through the translocator channel, as shown here. So that's just a general idea of how people think this translocator might function. And of course, it goes without saying, this translocator needs to be closed if there's not a protein being actively synthesized, lest any material that's in the lumen of the ER, like stored calcium, for example, might leak across the membrane into the cytosol. We showed what might happen for a soluble protein. The signal peptidase cleaves the signal sequence off, and you've got a soluble protein. What about the insertion of a single pass integral membrane protein? Well, we've got the same principle involved, but remember I told you earlier, consider hydrophobic sequences as a start and a stop transfer signal. So here is a start transfer signal. It binds into this translocator. Translocation is occurring as translation is occurring. So you've got a loop of protein that's being translocated through the ER membrane as protein synthesis is occurring. But now you suddenly encounter a stop transfer signal. When this stop transfer signal is encountered, perhaps this protein is released from the translocator. Perhaps the signal sequence is cleaved off, but the translocating protein is released when the stop transfer signal occurs. Synthesis continues on the cytosolic side of the membrane, and here you've got a transmembrane protein with a domain on the luminal side of the ER, and in this case it would have to be the N-terminal domain, and the C-terminal domain sitting on the cytosolic side of the ER. This is a single pass integral membrane protein. Here, rather than having the signal being clipped off right at the N-terminal end, suppose you've got a signal sequence, rather than at the N-terminal end, it's somewhere internal to the primary protein sequence. So this N-terminal amino acids are being synthesized on free polyribosomes. Suddenly you encounter this hydrophobic start transfer signal. This is bound by a signal recognition particle. Translation is paused. The whole thing attaches to the ribosomes attach to the ER membrane. Translation resumes, and you're resuming translation and translocation. The protein is moving through the ER membrane. Then a stop transfer signal is encountered. This thing is expelled from the translocator, so you've now got a protein embedded in the membrane, N-terminal end in the cytosol. This loop portion is in the lumen of the ER. Translation continues because the protein is kicked off the translocator, but now this C-terminal end stays in the cytosol. What do you've got? You've got a double pass integral membrane protein with a luminal domain and a cytosolic domain as a result of start and stop transfer signals. Look at one like this. Multiple start-stop transfer signals can explain how you get multi-pass integral membrane proteins. And as a matter of fact, you can do what are called hydropathy plots and predict based on the amino acid sequence of a protein, or likewise based on the messenger RNA sequence for the messenger RNA that is going to code for that protein. You can predict how many times a protein might span a membrane. This is an example of rhodopsin. You don't have to remember that this is rhodopsin, but rhodopsin is a light-sensitive pigment in the rod photoreceptors in the retina. It passes 1, 2, 3, 4, 5, 6, 7 times, a 7-pass integral membrane protein, and it can be predicted based on the primary amino acid sequence. It can be predicted that it would be a 7-pass integral membrane protein. Multiple start-stop transfer signals can account for this orientation of the multi-pass integral membrane protein.